Okay, let's dive straight into our genes today. It's kind of mind-blowing when you think about it, the sheer scale of human genetic diversity. Millions of tiny single-letter changes scattered across our DNA.
Yeah, millions. And a really big chunk of those are what we call missense mutations. So that's just one DNA letter swap leading to one different amino acid in a protein chain.
Like a single typo changing one word in a crucial instruction manual.
Exactly. It can sometimes, have no real effect, or it can completely change how that protein works.
We see dramatic examples, right? Like the single change causing sickle cell anemia. Tiny cause, huge consequence. But the problem is we found millions of these variations in people,
Mm-hmm.
and figuring out what each one actually does using standard lab tests, it's just incredibly slow and expensive.
Totally. The wet lab work is vital, for confirming things, but it just can't possibly keep pace with how fast we can sequence genomes now.
Which leaves most of these mutations stuck with that label. What is it? Variants of uncertain
Yeah.
significance.
Exactly, VUS. And that uncertainty is a massive roadblock. Think about trying to diagnose a rare disease or deciding if a specific drug therapy might work.
We need a faster way to sift through them, a shortcut.
Precisely. And that's where computational tools, especially machine learning, are really starting to make a difference. They offer a way to screen these mutations much, much faster.
Which brings us neatly to our source material for this deep dive, a paper introducing a new algorithm built for this exact task.
Right. It's called XGBMut. And its whole purpose is to predict if a missense mutation is likely to be, well, basically harmless, neutral, or potentially harmful, what we call deleterious.
Okay, so XGBMut, let's unpack this. What kind of information does it actually look at to make these predictions? How does it work under the hood, so to speak?
Well, it integrates quite a bit of data about the protein context, things like where the mutation actually happens in the protein structure. Is it in a region known to interact with other molecules?
Okay.
Or is it, for instance, in what's called a disordered region?
Hold on. Disordered region. What exactly does that mean for a protein?
Good question. So many parts of proteins fold into very specific, stable 3D shapes. But these disordered regions, they don't really have a fixed structure. They're more flexible, kind of floppy,
Oh, okay.
and they're often really important for, say, binding to other proteins or DNA. So a mutation there might disrupt function in a different way than one in the stable core.
Right, so location and local structure matter a lot. What else does XGBMet consider?
A really big one is evolutionary conservation. It looks at that specific amino acid position in the protein and asks, how much has this position been preserved across different species over, millions of years of evolution?
So if nature hasn't changed that spot in ages across many species?
It probably means that position is critical for the protein's function. Messing with it is more likely to cause problems. Like a part in a machine that's been the same forever, probably best not to swap it out.
Makes intuitive sense. Anything else?
Yeah, it also looks at the specific physicochemical properties, like does the mutation change the electrical charge or the size of the amino acid significantly? And it considers how likely that particular swap is based on known patterns.
Okay, so it's pulling together structure, location, evolutionary history, chemical changes.
Mm.
Quite a comprehensive approach. How did they actually train this model?
They use a machine learning method called Extreme Gradient Boosting, that's the XGB part, and they trained it on large, curated databases of known human mutations.
Databases like.
Things like Clinavar and Humsavar. These are big public repositories where researchers and clinicians report links between specific genetic variants and observed health outcomes like diseases.
Ah, so it learns by looking at thousands of examples where we already know if a mutation is benign or disease-causing from clinical evidence.
Smart.
Exactly. Learning from the ground truth we have.
And one thing that really stood out in the paper was the focus on usability.
Yes, that's a huge plus. Many bioinformatics tools can be tricky to use. You might need to set up a server or run complex commands.
But XGBMut was designed with a graphical user interface. The idea is you can run it on a standard computer without needing deep programming or bioinformatics skills.
That opens it up to way more researchers, maybe even clinicians
Mhm.
directly. Okay, the bottom line. How well does it actually perform? Did it work?
Based on the paper's evaluation against 10 other common tools, it did very well. They reported an accuracy of around 82.8% on their test dataset.
How does that stack up?
That was competitive, actually better than several well-established tools like SIFT and Polyfin2 in their specific comparison. But accuracy isn't the whole story, is it?
No, you also need to know how many mutations it can actually give you an answer for.
My coverage.
Right.
Absolutely crucial. Some tools might be slightly more accurate on the mutations they can analyze, but they fail on a significant portion. The paper highlighted that XGBMut achieved basically full coverage 100% on their test set.
Wow, 100%.
Yeah, contrast that with, say, another tool they mentioned, PONP2, which maybe had marginally higher accuracy but only provided predictions for about 57% of the mutations.
So XGBMut gives you an answer for pretty much every missense mutation. And that answer is right about 83% of the time in their tests. That sounds like a really practical balance for screening.
Exactly. That combination of solid accuracy and complete coverage makes it really useful for that first pass, trying to figure out which variants warrant a closer look.
Okay, let's try to sum this up then. XGBMut. It's a computational tool using machine learning,
Specifically, extreme gradient boosting.
right, to predict whether these common missense mutations are likely neutral or harmful.
And it does this by looking at key protein features, things like interaction sites, those disordered regions we talked about, and crucially, evolutionary conservation, all trained on known mutation data.
Performance-wise, it looks good, around 83% accuracy in their tests, which is competitive. And importantly, it gives you a prediction for essentially all mutations, 100% coverage.
Plus, it's designed to be user-friendly with a graphical interface, making it accessible beyond just bioinformatics specialists.
And you can really see why tools like this are so important. They let researchers and doctors sift through that massive list of variants much faster.
Definitely. It helps prioritize which ones might actually be driving a disease, speeding up diagnosis, potentially guiding treatment choices. It's a huge help in bridging that gap between sequencing data and clinical action.
But here's a final thought, something the paper itself touches on.
Hmm.
Even with a tool this powerful, getting around 83% accuracy, that still means a significant number of mutations are misclassified.
Mm-hmm.
Some harmful mutations might be predicted as neutral false negatives. It's just a stark reminder, isn't it, that even with these sophisticated algorithms predicting the full biological impact of every single genetic tweak, well, that's still a really complex challenge we're actively working on.